Good afternoon, and welcome to the American Floral Endowment's Grow Pro webinar series. I am your moderator, Olivia Liebing, and I am a current graduate student at The Ohio State University studying the propagation of ornamental woody nursery crops. Today's session is on plasma-activated water for floriculture production. On behalf of the endowment, I'm excited to be a part of AFU's Grow Pro webinar series that features a new topic every month presented by an industry expert. Dr. Nansen, welcome and thank you for presenting today on plasma-activated water for floriculture production.
Well, thank you for that introduction.
Recording in progress.
I'm here to talk about plasma-activated water. Probably something a lot of people haven't heard about, but I hope after this session that this is something that has interest, something you can find relevant to your crop production. You're being kindly asked, because this is such a new topic, I am kindly asking you to take your phone and point it and scan this QR code, because that will give us some feedback when we get to the Q&A session. So please scan this code. You don't need to register, but just put in some keywords, something that is related to why you are here today. We are hoping to make this the QA session more relevant, more interactive by you contributing. So the keywords that you're providing will then go into a word cloud, and that will give us something to talk about at the end. So please do this before I move on. All right, and before we actually get into the plasma activated water, there's a couple of details, very, very important details that I want to share with you. And that is, one of them is related to how plants uptake nitrogen, right? So most plants, unless they are leguminous, where they have interaction with mycorrhizal bacteria, the vast majority of crops, they uptake nitrogen either in the form of ammonium or nitrate, and that can, of course, there can be precursors of that. But why this is important is because fertilization is, of course, a big part of growing a healthy crop, whether it's a flower, whether it's an ornamental plant, or whether it's food. And a lot of that actually ends up getting into the atmosphere either as nitrogen or as ammonia. And that means that we have to really think of nitrogen and nitrogen management as a budget that is very dynamic, that is related to what goes on in the soil with the abiotic conditions. And there's a big chance that we won't actually give our crop enough of the nitrogen that it needs. So when we talk about crop production, not only flowers, but anything else, it is extremely important for us to think about where is that nitrogen going to come from as the population on the planet is growing or staying the same, but we overall have an increase in health and therefore also an increasing demand for crop fertilization. We can essentially look at the challenge of population growth or wealth on the planet as where is the fertilizer going to come from to produce all that food that we need and all those crops that we need. And what is ironic is that we have an atmosphere that is full of nitrogen. It's just not in a form that plants can uptake. They cannot directly uptake that N2. But actually, if we look at how much nitrogen there is in the atmosphere as a whole, if we were to take all that and turn it into nitrogen fertilizer, we would have fertilizer for 38 billion years based on the current use of nitrogen fertilizer. So what I'm saying is we are breathing on every day, every second of our life, we are breathing in, and we have right around us, we have a source of nitrogen that we could potentially turn into use in our crop production. We just have to use the right technology. So where is that? What technology are we going to be using? Well, that's where plasma activated water comes in. And you're looking at it right here. This is us in the lab generating plasma activated water. What you're looking at is a jet that is inserted into water, atmospheric air is being pumped in through the jet, and it then gets subjected to a very high electric charge that breaks those bonds in the atmosphere. So remember, we have about 12% oxygen and we have 78% nitrogen, or somewhere along those lines. And those O2 oxygen molecules and those two nitrogen molecules, because of the electric charge at the tip of this jet, they get taken apart and then they get blown into water, and then they react with water, and that creates all what is referred to as reactive species. It does so now we have three free nitrogen that can actually work with the different oxygen molecules and the hydrogen molecules, and they are very, very reactive, and that creates a myriad of species in the water, and that is precisely what we are trying to take advantage of. So before we get back to plasma activated water, I just want to mention that what I'm going to be talking about is obviously part of a team. So these are the team members that are part of my team here at UC Davis. And you will be seeing some of the work that they have been doing. But we are also part of a much bigger team. We are leading a consortium of people from Minnesota, Cornell, UC A&R, and also from Ag Engineering here at UC Davis. And we hold a wide variety of expertises, and that's precisely what we're trying to put together as part of this work into how we can make plasma activated water a sustainable nitrogen source, and also use it in other ways to promote sustainable crop production. And we are extremely grateful to the funders that have provided us with the resources to be able to do this. And I think in the current environment, funding environment, where basically virtually all funding to universities has been put on hold, I think it's very important to recognize that what I'm sharing with you today would not be possible had AFE and some of the federal and state funding bodies, had they not been available. We would not be able to be here today. We would not be able to show you some of the research that we are doing. So this is very, very important. And I want to take that a couple of steps further by saying that we feel that we are part of a very, very large and very, very wide body of researchers that are truly committed to trying to address the needs of the future. We are trying to develop the solutions. We are trying to educate the young people out there so that they have skill sets and are able to go out and be able to, either through means of policy or but also in research, develop the solutions that are needed for the future. So this is very, very important that this is highlighted. Anyway, back to our main player, the PAW, the plasma activated water. So in this process, depending on how long we treat the water, we change the pH, we create nitrate, which is NO3. So the nitrogen in the air can go together with 3 molecules of oxygen in the water and also in the air and suddenly create nitrate. And we change the conductivity of the water. We change the oxidative reactive potential. A lot of characteristics of the water are fundamentally changed, and it will depend on the characteristics of the water we start out with and how long we treat it. And it's also very important to remember that we can actually treat it for too long, kind of like with a if you apply a pesticide if you apply it to a crop if you apply it at a too high dosage, it can cause phytotoxicity. Well, the same thing with PAW. It can essentially be too strong, and that will have negative effects. So there's some tweaking, and there's not one kind of PAW. There's going to be different PAWs for different crops, for different applications, for different soil media. It's a big area where there's a lot of research that needs to be done. So we are using it particularly to boost the germination of seeds. This is something we are learning, that it can speed up the of seeds that have a lot of dormancy, that have a thick seed coat, that are where priming is typically needed, and where maybe some acids or something are being used that is kind of dangerous to work with. Maybe PAW could play a role there. We are seeing that seedlings can grow much faster if we're using the right PAW, and more uniformly, which is very important in crop production. If you can get your crops through those initial life stages where they're very susceptible to diseases, and to insects, if you can get them through the production very quicker and more uniformly, so robots can pick up the seedlings, that's very important. We are also testing it as fertilizer down the road, and I'm going to show you some pictures of that. And then in pest management, I'm going to share some slides with you about that. We are trying to use it as a way to boost the plant's own resistance mechanisms, their ability to recruit natural enemies. And we're even looking at it as a way of, instead of using regular water, maybe using it as the carrier in pesticide applications. So some of these aspects will be dealt with in this presentation, but this is something we can also follow up on in the future if there's an interest in this, where we can provide even more details about the results we're getting. We have recently, so this is Pamela. She's an undergrad. She will join my lab in the fall. She's the first author on a paper we have recently published, where we talk about this plasma activated water as a sustainable source of nitrogen fertilization, and how it meets some of the sustainable goals that have been put forward by United Nations. So this is sort of on a broad level why we're doing this. So if you're interested in learning more about the bigger picture, and also we talk about the Haber-Bosch process and why and how this is more sustainable. Of course, use of the right kind of energy it is very energy demanding, because we have to create that electric charge. But if you have, the sustainability depends on having access to renewable energy. But we know that solar panels and windmills and other sources are becoming more and more important, and are actually, in today's world, producing energy that is cheaper and way more sustainable than fossil fuel derived energy, which the Haber-Bosch process, producing synthetic fertilizer, is based upon. So there's a big advantage. But have a read, have a look at this. This is freely available. Then Patrice has produced a series of papers, and there are more in the pipeline, about the pest management and how this is affecting critical pests. We are focusing on spider mites, but also western flower thrips, how these pests are they respond very differently to plants that have been irrigated with plasma activated water, so through supplementary irrigation. And we're learning through Patrice's work, we are learning more and more about the mechanisms involved in this, and finding out why this is happening. So this is also something you can tap into if you're interested. We have a website where we compile all the information. So there are some past meetings we have held. There are some newsletters. There are different presentations. So this is a continuing compilation where if you're interested in cold plasma and learning more about what is going on, we recently held a summit, which some of you may have attended or seen an invitation to. If you didn't get a chance to see that, there is a YouTube link here on this page. So this is also where you can meet some of the industry players, some of the researchers that I mentioned on that previous slide from Minnesota and Cornell, and from other universities that we invited. So industry people talking about a whole bunch of aspects. There's like 15 presentations compiled in that virtual summit. So feel free to join that. That's also free. So with that, let's talk about how it can actually affect growth of plants. And you are looking at 3 tomato plants here where they were planted at the same time in the same kind of soil in the same greenhouse, irrigated the same way and fertilized the same way, except the 2 to the right were given, instead of given regular tap water, they were given about 40% of the water they were getting were 2 kinds of PAW. So they were getting some more fertilizer. So you can probably, it's probably fair to say that the one to the left was under fertilized. So that's why it's a little bit yellowish. But you can see here the dramatic difference that there is definitely fertilizer in the PAW. Now we think it's actually more than the nitrate that is being provided from the PAW. There's a lot more going on in interactions with microbial organisms. There's a lot, and you can also see it here with strawberries, that the same thing happens, that strawberries also responded very strongly to this, right? And we have marigold plants, so getting into the flower production, we are definitely seeing also that these plants, that across the board we are seeing, unless they are leguminous, we are not seeing the same response. But if there is something going on, and there's a lot to be learned about exactly what is driving this increased growth without having it doesn't seem to have any negative impact. And so the question really, of course, becomes how can this be applied? How often does it need to be applied? In what concentrations? At how frequently? What is it going to cost? Those are the downstream questions that we are hoping to engage with you as growers and others. We are hoping to figure out how we can do this together, so that these solutions that we are researching, how we can make them feasible to you. Here what you're looking at is, and we're talking about pest management. We're looking at what happens if we experimentally take some leaf discs that are collected from some plants, and we are either irrigating them only with tap water, or if we are supplementarily giving them some PAW. Then we take those those leaf discs, we put them into a petri dish, and we put some spider mites in between the two. So we're essentially giving them a choice, and we then count where they are and where they lay their eggs. And you can say, well, this is very experimental. This is very artificial. It is, but it also means that we're simplifying the universe, and we're trying to assess the bioresponse of the individual pests, because if they're showing a difference, then it becomes very interesting as trying to figure out why this is happening, right? So the results that Patrice got in these preference or these choice bioassays are illustrated here in this bar. So you can see that the red ones, when we give them a choice in the top between a control and a PAW that has been treated for 6 minutes, that's why we call it PAW 6.0, you can see that they prefer to be on the control. The red bar is bigger than the blue bar, as you can see right here. You can also see when we give them a choice in a separate set of bioassays, when we give and we run this for a lot of times, you can see that they are basically, when we give them a choice between PAW that has been run for 9.4 minutes, we see the same effect, that they basically don't like it. They don't like the PAW treated plants. But if we give them the choice between the two PAWs, there's no difference. There's no significant difference between the two. So there's something about these leaves, even when they have been taken off the plant and stuck into a petri dish and given 48 hours, Why is that? And even further, when we then do what's called a performance test, when we then take the plants and we don't give them a choice, but we just supplementarily irrigate the plants either with regular water or with control plant, look what happens here. If we follow them for 3 weeks, we can see that the population growth on the PAW treated plants reaches about an average of some maybe 55 or somewhere in the 50s, while on the control plants we were twice as high. So the development rate of these pests, of these mite populations, was much faster and much higher on the control plants. So not only did they, so this one right here showed they don't really like it when they have a choice, and this one is now showing when they have no choice, they are not doing particularly well. They simply just don't develop as fast. And see, this is very important, because of course, if we talk about thresholds and if we talk about use of biological control agents, if we can slow down the development of the spider mite populations, maybe predatory mites will have a much better chance at controlling our pests. And maybe we can, if the growth rate is much, if it's much lower, if they don't develop as fast, maybe we can get the crop out of the greenhouse before it actually reaches a threshold, before it actually becomes a problem. So by slowing down just by slowing down the development of the spider mites, it opens up a lot of alternative applications. We may also look at organically certified pesticides that may or may not provide very super good control if we have high populations, but maybe they work when we have lower populations. So maybe this can be used as a way to also improve the performance of softer chemicals that we otherwise wouldn't consider, simply because we are afraid of these spider mites growing out of control. So when we do something very similar with Western flower thrips, which is what we're looking at here, and this is what a visiting scientist from China, Ning Li, what she's working on. So we are basically seeing the same thing, that they are, again, when we expose them to choices, they do not like them. So we are expecting, just like with the spider mites, that they are also avoiding that they are also not in that they will develop much slower and give aureus and other natural enemies a better chance of performing well. So it's a way, as part of integrated pest management, we really see the PAW as a way to boost other mechanisms, other elements of a pest management strategy. And another aspect that I want to talk about today is that we are looking into, and this is Yu Gong, who's a master's student in my program. What he's looking at is he is basically looking at the stability of PAW over time. Because what we are also learning, as I told you early in the very beginning, we break these covalent bonds between O2 and N2, and that makes them very reactive. Well, it turns out that when we create this PAW, it actually turns out to be very reactive, even weeks after it has been generated. So we need to on one hand create these chemical reactions, but then we also need to stabilize them at some level, so that we know what product we are applying. Because if it's kind of like going to the shop or going somewhere and buying a product, and then 3 days later it's a completely different product, that's not desirable. That becomes a problem. So what Yu Gong is looking at is figuring out chemically how we can stabilize it, and how the storage conditions we are using, how that can be used to create an environment that is as stable as possible. So what we are looking at, and we're working with chemists, and this is where it gets very multidisciplinary, the specific experiments that he's looking at is whether we close the bottles, which means that we fill them up all the way and then we seal them so that there's no head space or very, very little head space above them, above the water in the bottles, or we keep them open, which means that the plasma activated water can react with the atmosphere. So we're looking at whether that's a problem, because if it's a problem that there is a headspace or there's access to atmosphere above, if there's a lot of exchange going on right at the surface of the water, then we need to, when we produce it, we need to seal it, and then we need to keep it sealed until we use it, and then use the entire bottle. Kind of like if we know with some pesticides, if we leave them in a bottle, they will deteriorate over time. We should ideally use everything at the same, especially if it's a biological product. Well, this is what we are trying to figure out with PAW, how reactive it is. Another thing that we are looking, that Yugong is looking into, is the use of bases. So particularly we are looking at because the PAW, as we treat it, the pH goes down dramatically. Can we bring it back up? If we treat it depending on the volume of water, but we can, even if we take a liter of water, we can bring the pH down to 2 very, very within minutes. Well, that's very, very acidic, and plants typically want something in the 5 to 6 pH range. Well, can we use a base to then bring that up? And could we potentially use a base such as potassium hydroxide? Because then we would also be providing the potassium. If we have the N and the P, that would already be, and the K, sorry, we would need would be the phosphorus. So we're providing 2 of the 3 elements. So we are looking at that as part of developing solutions that are meaningful, that are applicable to the commercial industries as part of this. So even though I am in the entomology department, you can see how far this is from entomology. But at the end of the day, if we are figuring out that this has profound implications for how we can develop sustainable pest management strategies, well, we are willing to also look into the chemistry of water and the physics of these cold plasma systems to make them as efficient as possible. That needs to be part of the equation, and that's why we need these multidisciplinary teams, and that requires money. So all of these elements need to be in place for us to be able to have conversations like this. So looking at, so sort of getting towards the end here, where what we are really trying to, as we are learning more and more about this technology, we are trying to figure out what is the best PAW for a given crop, for a given seed, for a given development stage. How do we store it? How do we process it so that it's storable for as long as possible? If it's not being produced right at the production site, and I should point out, if you go to that YouTube video, you will see that there are commercial systems already available that can tap into existing irrigation systems. So it is commercially feasible. When do we have to apply it in different development stages? Is it at the seed level it's most critical? Is it at the seedling stage? How should we apply it? Can it be sprayed onto the leaves without causing aesthetic, so phytotoxicity? Does it have to be, how does it go through drip? How much do we need to apply? What effects does it have on growth medium in a hydroponic system? There are so many questions here, right? And legal issues, which I'm sure some of you might have. Is this going to make my crop? Can I still export it? What can I sell it as? And in the YouTube video that I linked earlier, there's a specific presentation about the legal issues of PAW. It is unresolved. I can share that with you at this point. So there are still some legal battles. There's still some legal work that needs to be done. But this is also where we, as researchers, growers, as consumers, we need to put pressure on the politicians, on the bodies that are responsible for this, so that they can make this technology available to us, so we can produce fertilizer more sustainably. The economics of this, of clearly we have a small system in our lab. Well, that's not the most efficient way of doing it, but upscaling, how can we make it available? How can we scale it up? And that, depending on the demand, of course, this will change, just like with solar panels and with hydrogen vehicles and electric charging stations for electric vehicles. As the demand goes up, the price, the availability, the user friendliness, all of this will go in one direction only. But I think this is such a promising technology, that it deserves more attention, and it definitely needs more funding. It definitely needs more people to have a look at it, and we need to seriously consider it as one of the Lego blocks that we need to look into in the future as part of sustainable crop production.
I just want to show you, this is what we ended up with here. Kind of an impressive number of, or cloud of words here. And of course the size indicates the relative importance. And now people are maybe putting in even more here. So I don't know, Olivia, how you wanna run this, what you think how we should do this, but we can definitely see that health and innovation and pathogens are keywords. It's very, one thing that I did not mention in the recorded presentation was that one of the other chemical species that is being generated is not just nitrate and nitrite, but also hydrogen peroxide. And we probably all know that we can go to the pharmacy or elsewhere and we can buy hydrogen peroxide as a disinfectant. And hydrogen peroxide is also being used as a fungicide. Well, there are definitely people looking into use of PAW as a fungicide. And this has a lot to do with the hydrogen peroxide that is being generated. So there's just a lot of chemical species, kind of chemical compounds that trigger hormonal responses by the plants that have all sorts of features that we're just we're at the very forefront of, and the number of people now tapping into PAW as a research topic is growing quite quickly. So if you are into Google Scholar and want to look at papers, if you just put in plasma activated water, you will find lots of papers being published all over the world. So we are certainly not the only ones, but there's it's a very exciting area.
Yeah, it seems like this has got us getting a lot of great questions coming in.
Okay.
I'd like to start with, will plasma activated water assist with insecticides used in the extermination industry?
So in the extermination industry, so is this what does can the what is this extermination industry? So is this like in households and is that maybe so is this like pest control in households and commercial accounts? Is that what's being mentioned or referred to, you think?
I think that's what it is, yeah.
It's so it's definitely possible. I'm not aware of any applications. I have a background actually in commercial pest control, so I'm aware of that world for cockroaches and others. I don't know honestly where that would fit in, but it's possible. I yeah, I don't know. But certainly it's already being used in the medical field and in the hospital worlds for sanitizing, so surface sterilization, and the the computer industry is using it also to surface when they have components that need to be very, very clean. They also use different kinds of plasma, maybe not plasma activated water, but they use they basically, instead of blowing the plasma into water, they blow it straight onto surfaces, cool it down first, and that will basically, because these chemistries, these chemical species are so reactive, they will break down everything. So it's very corrosive. But if it's controlled, used properly, it's a very effective surface sterilant. So for surgical equipment and electronics and whatnot, it's being used quite widely.
And then another question, if a plant is treated with plasma activated water, does it reduce overall plant stress? Let's say the crop is outside in full sun with heat stress.
So we are right now, what I was trying to convey, and maybe I wasn't successful in that, but one of the things that I think is important for us to remember is that there's not just cold PAW. Can some PAW cause stress in plants? Absolutely. We can probably kill plants with PAW. So could it be used as a herbicide? Possibly. That's actually possible. Can we induce stress? I would say yes. If the water that has been treated for too long. Now, where I want to take the question is, we are right now we're growing. So I'm in California, and in a month or so, we're going to be harvesting tomatoes. We treated some processing tomato transplants with PAW. So as they were in the greenhouse and getting ready before transplant, we treated them with PAW. We didn't treat them out in the field, but we hope that we can demonstrate that pre-treatment gave the plants, it triggered the plants to grow a root system that was stronger, better at adapting. So when they were transplanted out, they had less stress and therefore were able to start their new life outside much better. And that is relevant for ornamental plants that are being transplanted as well. Can we use PAW? So the person who asked the question was talking about heat stress. Well, can we make, I think another way of maybe modifying the question or taking the question in a particular direction, can we make ornamental plants more resilient to heat stress using PAW? And I think potentially the answer is potentially yes, that we can, there are already published papers demonstrating that the root structure of plants that have been irrigated with PAW is very different, and therefore the plants are better at taking up nutrients and finding water and whatnot. So I think in that sense, I would say we can mitigate some of the climate challenges we are facing using PAW.
Were you measuring the ORP of the PAW? If yes, what was the ORP of the water that was applied to the plants in the spider mite experiments? And how was it applied?
So we were measuring the ORP, and it's in the paper. I can't remember the exact numbers, but the person can look up the papers, and we can definitely and also feel free to reach out to me afterwards if this person wants more specifics. But yes, we were measuring. We are measuring a whole bunch of diagnostics of the water. Absolutely. Conductivity is another very interesting aspect. So think of it as a seed. If my hand here is a seed, if the water has higher conductivity, it's able to bind to the seed and to the seed coat and penetrate in through the seed coat and reach the embryo more quickly. So if it's carrying nitrate and other nutrients with it and moving faster into the seed coat, maybe that's why we are seeing a faster and more uniform germination. So it's not just ORP, it's all of these characteristics that, and as it's penetrating further in, if it has hydrogen peroxide and other elements, other components in it that trigger hormonal responses in the plant, well, then there's just a cascade of things happening inside the tree, inside the seed. And that's the fascinating, we know very little about what's going on, but we seem to be seeing effects. Not always, and it's not with all seeds, but in some seeds we're seeing some very interesting effects. If you go to that virtual summit that I referenced, if you're all ornamental growers, this may not be interesting to you, but some people may know that spinach is a crop that is notoriously difficult to germinate. Well, we have obtained some very, very interesting results with my colleague at Minnesota is showing in that video some very promising results with spinach, a challenging crop. And I'm sure that in the ornamental world, you have a lot of seeds that are funky, that are difficult to germinate. Well, maybe we can help you with that.
And then would you mind going into what kind of changes were observed when plasma activated water is stored, and how long were you storing it?
So we are still, so the pH changes, all the characteristics that we were measuring, all of them change, but we overall are noticing that if we after the water has been treated, if we bring the pH up to a high, if we make it more alkaline, we can stabilize more. We can generally stabilize all of the characteristics. So we are promoting the use of potassium hydroxide as a way to do this. But we want to look more into this. We want to look at also other bases. We think that this is sort of a first. We have also looked at sodium hydroxide, but we don't want to put sodium into. We already have problems with salinity of our water, so that's a problem. We need to work with we need to work more with chemists, and we need to work more with growers to figure out what exactly is it that what bases should we be using, which ones are most available, are safe to work with, cost effective, and seem to give the best bang for the buck, basically.
And this one kind of follows up. Zane was asking, how stable is plasma activated water over time?
Right. So this is what Yugong and this is something we're tapping into. We when we initially got our grants, we were not quite prepared to the dynamic, how volatile, how reactive this process is, and how it continues to go. I think the previous question was not. There was one aspect I didn't mention. We are looking at PAW for months after. So we have some samples. We're going to store them for a full year and see when they finally stabilize. It's very reactive, and it keeps reacting, and that's why it's a very important aspect to make sure that we learn as much as we can. So it at some point it's going to be stable, but there are a lot of variables that play into this. Another one that maybe I mentioned it, but it maybe got hidden in there is, what is the quality of the water that you start out with? And we are looking at different water sources and see how that plays in. So maybe you have to even pre-treat the water before you subject it to cold plasma. So there can be some pre-treatment and some post-treatment. And this is where it becomes kind of like the adjuvant business of pesticides, where you have the process or the chemical itself, but you have all sorts of other aspects. And we are basically creating market opportunities here where young people can come in and start businesses. If we can store it properly, well, then it can be produced somewhere else and then be sold to production units. But exactly how is this going to be done? What are the diagnostics? Who are going to be doing the diagnostics? There's a lot of exciting job opportunities in in the future that can come out of this. And this is something we're very excited about.
And the next question is, does plasma activated water reduce algal or moss growth in potted plants, since hydrogen peroxide is also produced in generating plasma activated water?
I could definitely see that. I would suspect that. So again, if the goal is to kill stuff with it, absolutely, you just crank it up. You can definitely kill stuff with it. It absolutely. It's used as a surface sterilant. So you can, of course, you don't want to negatively affect your crop. So it's got to be a balance there, but absolutely.
And then this question's from Steve. Is there a disinfecting action to the irrigation solution? I noted the ORP increase mentioned, and I'm curious if this can reduce pathogen load in the irrigation system.
This is so just like UV light is being analyzed. Again, remember that I'm in the entomology department, so I'm way outside where I'm supposed to be, but I'm just sharing my experience anyway. So I know that in the hydroponic systems where they are recycling using the same water, many of the systems are running their water through a UV light to sterilize the water. Well, the same could potentially, the PAW could be having the same function, because it's definitely killing pathogens. Absolutely. So there's ways that the hydroponics, so plasma activated water and hydroponics go very well together. And I'm sure that there are, if you YouTube or you Google search that somewhere, you will find people who are in that space for sure.
And this question's following up again on the spider mites. How is the irrigation applied when controlling spider mites and thrips?
It was applied to the soil, and I kind of left out some of the mechanisms that we looked into, but I can share a little bit more if people are interested. So what Patrice demonstrated was that the trichome density on the tomato plants that he tested significantly increased. Now, people who, so trichomes are these small hairs, and they can be glandular or non-glandular, but so they are a physical barrier for pests that move around on the leaves, and the glandular trichomes actually contain compounds that are toxic to the herbivores that are on the plants. So it's very interesting and explains our results quite well, that the trichome density was increased by the PAW, and therefore that's why they preferred not to stay on those leaves. Now, trichome density in some plants is very important, like cannabis and others. That's actually what growers want. So could this be used as a way to boost the quality of a crop? Absolutely. I in my program, we don't we don't work with cannabis at this point in time, but I'm sure other people out there who are would be interested in this. And there may also be other ornamental plants where trichome density is important. I don't know of any, but that's one part of the explanation, is that the plants it boosted this defense, this important aspect of the plant's innate defense. Also, it changed the element composition a little bit. Yeah, so there are some mechanisms there. We also hope to demonstrate in the near future that plants' ability to recruit natural enemies, so the volatiles, that the inducible defense compounds that plants produce when they get attacked by herbivores, maybe that ability to signal and recruit natural enemies is being boosted by PAW. That would be a very interesting result. So we're looking into that.
Great. And have you looked into the changes of gene expression comparing controls versus plasma-activated water treated plants, and which genes are upregulated by plasma-activated water treatments?
That's where I will talk about UV. I will talk about some of the physics and the math behind it. Molecular work, that's kind of where I've just decided I don't know. If somebody is interested and knowledgeable about this, please join us. Please reach out, because this is an area. There's a lot of, I mean, the seed physiologists need to help us figure out what happens at the seed level. The molecular people need to help us understand which genes are down and upregulated. Absolutely. There are other programs working on that. We are just, that's kind of where I'm drawing a line there. That's too deep a pool for me to go into.
So here's another question from Zane. What is the long-term impact of irrigating crops with plasma-activated water on soil microbiota and fertility?
We have no idea. We have absolutely no idea, but we are very interested in that. There's a growing interest in biostimulants and a growing appreciation for soil health. We think it's very important to look at how PAW into how that can actually be a player. Will it, we if it kills stuff, then it could potentially kill some of the symbionts. So is it having a positive? Is it having a negative? Is it what is going on in which soils? Which microbes are being affected disproportionately more by PAW? We don't know. This is an area, long-term studies need to be done. Let me just say that PAW, so activation of plasma of water, has existed for a long time, and the technology of using plasma, of course, is not new at all. It's just that it has become more user-friendly, it has become more cost-effective, and there's a growing need for alternatives to fossil fertilizer or synthetic fertilizer and whatnot. So the technology has been there, but we don't have those long-term studies yet to answer that very important question. We need land-grant universities. We need resources to set things like that up. So yes, absolutely.
All right, well, if everybody would like to keep adding questions into the chat or Q&A, I have one I'd like to ask myself. Are you looking for grower collaborators to participate in studies to test PAW?
One of the reasons why I'm here today, one of the reasons why we're doing all this outreach is precisely because we are deeply committed to finding solutions for the needs in the future. And we are not just sitting as scientists in our offices and crunching numbers. Please look me up. You're very welcome to email me directly and say, hey, Christian, I saw your talk, blah, blah, blah. We grow, blah, 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 and let's have a chat. Let's see what, and because we have this consortium with people in different places around the country, and we will, at some point, there is going to be an opportunity to write grants. We will need places to do commercial validations. So when the grant opportunities open up again, we will definitely be very keen to reach out and engage with commercial enterprises in testing of PAW.